Early on, it seemed clear that the linear sequence of bases in a gene must somehow dictate the linear sequence of amino acids in a protein.
And I personally thought that this was the key problem. How do you get from that sequence of bases to the sequence of amino acids?
In 1957, Francis Crick teamed up with a new scientific partner, Sidney Brenner. Their challenge was to find out how genes produce proteins. They actually had two information problems to solve. The first one was transportation. Chromosomes are locked within the nucleus, while proteins are manufactured in the cytoplasm of the cell in structures called ribosomes. What carries genetic information from chromosome to ribosome? The second problem was interpretation. How does this message get translated from the language of DNA into the language of protein? Attention now focused on RNA, a molecule that might hold the answer to both problems. A single-stranded relative of DNA, it is found in both nucleus and cytoplasm, but more so in the cytoplasm. It was also known that RNA is involved in protein synthesis. So it was a good candidate to be the transportation agent from chromosome to ribosome. And it was also possible that RNA could act as the dictionary that translates the language of DNA into the language of protein.
In the first years after the publication of the model for DNA, there was already a group of scientists who had formed what they called the RNA Tie Club.
The RNA Tie Club was founded by a well-known physicist, George Gamow, who was intrigued by the double helix structure. There were 20 members, one for each amino acid. The last two members were Francis Crick and Sidney Brenner.
Francis was tyrosine and I was valine, so I just got in at number 20. It was regarded as a very fringe group, in my opinion, by, the kind of establishment science.
They speculated that genetic information flows from DNA to RNA to protein. Crick later termed this theory the central dogma. He chose the word dogma because there was still no experimental evidence to back it up.